the jungle woman thing!
This is the pilot year for the internship collaboration with Tirimbina Research Station in Costa Rica. We're fortunate to have 6 really wonderful interns. They're a good mix of strengths and interests and backgrounds. They also get along really well. We have somebody who's an agronomist from Latin America, so she's wonderful with the language and the plants. They're all microscopists, so they all know the microscopes really well, and they don't necessarily know how to be out in the jungle or how to collect specimens, and they're learning that because they're working with these great field guides and researchers from Tirimbina, so they're learning the jungle and what's out there. It's fundamentally an exploration of possibility, and I've been calling it a giant science party, actually. You know, we're just coming down here and we're going to feed off of each other, and all the scientists here have already been throwing us really interesting ideas about things to look at under the microscopes that would never occur to us because we don't know what's down here. That's the point. They do, we don't. But we have this great tool that they don't know about, and we're educating them and they're educating us. And because our department, through Hank Fabian, already had a connection with this wonderful research station down in Costa Rica, we thought we would merge field research out in the field, collecting things, good old naturalist, environmental science, where you really know the jungle and you know what you're looking at with your naked eye, and bring these scopes to the station and see a whole nother world unveiled. In this first year, we figured out how to transport 3 very delicate, precious microscopes across several borders, and we've been fortunate in being able to bring 3 of these microscopes down here, and also basically we set up a little bit of a cell and molecular biology lab plus a field lab. All of this equipment are things that we brought down with us in order to process the specimens and collect the specimens and be able to do the kind of work we do back home. Just put some mitotracker on that guy, which will stain the mitochondria. This one it's the same specimen, and I do 3 different stains. So this one will stain the DNA in one color and the mitochondria in another color. Okay. And if these plants are still, well, fungus is still alive, you can actually see the mitochondria moving around because they're alive.
Right.
Merritt Microscopy program takes 1 year. It's a very intense program, and the nice thing about it is that it welcomes people with very different backgrounds. So we started together this program. Actually, Peter was the one who found the information, the brochure at the college, and then he came to me and he told me about this program in microscopy, and I thought it was interesting, and that's how we got into it.
I thought this is a great marriage between both the science foundation I was looking for and the area of comfort I was with, which was software imaging.
Toby's gonna draw.
The microscope has a wonderful tendency to frame things in a way that are artistically pleasing, and the artist in me enjoys using a microscope to produce beautiful images. Definitely the program and microscopy itself, learning that tool will benefit anyone who's interested in science.
I just want to say one more thing about these scopes. These microscopes were created in order to do field research in regions where there isn't a lot of access to infrastructure such as electricity. They can be solar powered. They use a new illumination technique that allows them to go for over 10,000 hours without needing a new bulb installed. And we also have 2 very wonderful dissecting microscopes.
Closer.
All of these scopes are digital, so we're capturing digital images. So they're sort of our first processing station, and then we bring these specimens over here and look at them with fluorescence, which is this added value and interesting technique that is new to this area. One of the most exciting things about Costa Rica is its enormous biodiversity. In this research station, for instance, from the parking lot to the entrance to this building, they've identified over 200 species of birds. So that's just a few feet, basically. Costa Rica is a place where a lot of scientists come to learn about life on our planet because there's so much richness of different species of plants and animals and insects and reptiles and so on. The Costa Rican government wisely decided that the research had to be done in Costa Rica so that the economical advantages stay in the country. So there's a very, very strict law in Costa Rica saying that you cannot take any sort of specimen out of the country, not a leaf, not a flower, and certainly not an insect.
And I'm trying to take an image. It's very difficult when you have live specimens. Why don't you ask Manuel? He must know.
Me puede buscar.
¿Puede buscar?
Okay.
Métase Google.
Remember,
Sí.
then we can take the data that we find and compare it to literature or internet searches and so forth and try to figure out what species we have. So this is a great tool in the rainforest that helps you really bring what you find out in the rainforest, what lives out there closer, so that you can really see at magnifications that you can't see out there in the rainforest with your naked eye, and really tell what it is that you have.
And who knows what we'll discover these couple of weeks that we're here. We're very excited to see what happens.
For Alta TV, programs that matter.